picture shows an overview of the cell membrane. We're going to learn each component and what its role is in cell membranes. This slide discusses the overall presentation, and this is pulled from a AP Biology resource, but it does a really nice job of explaining membranes and their purposes. The key thing is that sets up a boundary, right? So the cell membrane is a boundary between the interior and exterior sides of the cell, and it separates living things from its non-living environment. Phospholipids are one of the key components of cell membranes. They're composed of two main parts. Here we see the phosphate head and the fatty acid tail, or over here we have a molecular diagram of the phosphate head and the fatty acids tails. The phosphate head is hydrophilic, right? So that means hydro prefix means water. Philic means it coincides or gets along well with, whereas phobic, you can think about phobias, the fear of, right? Hydrophobic is opposed to water. Hydrophilic works well with water. And so we've got this bilayer down here of phospholipid heads, which do well in aquatic environments, and a second layer of heads that do well in aquatic environments, and then this barrier established by the fatty acid tails, which do not do well in environments that contain lots of water. And a way you can remember that is that cats have tails, and if you've ever put a cat's tail in water, it hates it. It hates having its tail in water. So the fatty acid tails hate water. They're hydrophobic. Here we've got polarity related to hydrophobic and hydrophilic interactions. Water is a polar molecule, meaning that it has regions within the molecule that have partial charges on it. So to be hydrophilic means that you do well with polar substances. Hydrophobic do not do well with polar, but rather do well with nonpolar substances. So we've got this region, these three regions of the membrane: the outside, which is polar; the interior of the membrane, which is nonpolar; and the inside, which has the hydrophilic heads and is also polar. It's more than just lipids, right? Lipids are fats, and so there was a period of time where scientists believed that membranes were composed just of fat, but we've learned that there are also proteins that are integral parts of the cell membrane, so we'll talk a lot more about what those proteins do. So as a quick overview, we'll talk a lot more about each component. A glycoprotein is a protein that has a carbohydrate attached to its surface, and the main role of glycoproteins is cell-to-cell recognition, so that you can identify hormones can attach to specific cell types, and you can say this is a muscle cell or this is a skin cell, or you can also use this, your body uses this to determine foreign invaders. Pathogens that might cause diseases are going to have different glycoproteins than the glycoproteins on your cells. Phospholipids we discussed, they're the blue heads and the black tails coming down in. We have peripheral proteins on the inside or the outside. They stay on one side of the membrane. They don't span the entire membrane. So it's just this tiny little purple part down here. This bigger purple protein is a transmembrane protein, also called, so we've got some examples here, they're also called integral proteins, and they span the entire membrane, allowing substances to pass into and out of cells. We also have cholesterol in the membrane, which serves a role in the movement of these phospholipids. So the phospholipids aren't stagnant, they're actually fluid, and they move laterally, and they can also flip-flop from the top and the bottom of the bilayer. So cholesterol actually puts kinks in the membrane and changes the fluidity of the membrane. Inside we have these green wormy looking structures. Those are called filaments of the cytoskeleton, and they give the cell its overall structure on the interior side of the membrane. Then we also have glycolipids, which we'll discuss a little bit later in the presentation. So the amount of fatty acid and the type of fatty acid is going to change the fluidity of your membrane. Fluid membranes means that the phospholipids can flip-flop positions. So in a situation like this, where you've got fatty acid tails that have lots of kinks in them, those hydrocarbon tails or fatty acid tails with kinks have unsaturated fatty acids. That means that they're not completely filled up with hydrogen, whereas those that are saturated are more linear. They look straighter. they're not so crooked, and they have saturated fatty acid, which means that all of their carbons have maximum hydrogens attached to them. So the amount of fatty acids that you have on your tails of your phospholipids makes a difference in how fluid or rigid the membrane is. And so that's a kind of a higher level note we can talk about more in class. Membrane proteins, as I said, there's two types. There's peripheral proteins, which sit on one side of the membrane, and there's integral proteins, which span the entire membrane. When you're looking at proteins, particularly the integral proteins, they're going to have to mimic what we talked about previously, where you have hydrophilic and hydrophobic regions. So this polar section of the protein is this big squiggly thing. The dark purple section on the outside and inside is going to be polar, and the red section on the interior of the membrane is going to be nonpolar, which means that we've got a hydrophilic region, hydrophobic region, and hydrophilic region again to create a barrier and maintain that barrier established by the phospholipids. Some examples here, we've got an aquaporin, which allows water channels to allow water to go into and out of cells. And here we've got a proton pump, which we'll talk more about when we study photosynthesis and cellular respiration. This slide is pretty key. You want to understand these 6 main features or functions of membrane proteins. The first is to transport substances into or out of cells. The second is to serve as enzymes to catalyze reactions in cells. The next is to be a receptor, so a hormone can bind to a protein and alter what's going on inside of the cell. We have identification, which we talked about with glycoproteins, to say this cell is a particular type of cell in the body and it belongs here. Proteins also serve to hold cells to each other. So we've got a cell here on top and a cell on bottom, and the proteins are joining those cells together and attaching the interior section of the cell, the structural component called the cytoskeleton. You can have the cytoskeleton attached to proteins in the membrane. So the one that we focused on a little bit before, I talked about this already, it plays a role in the immune system, and we can talk more about this one in class. Let's talk about how things move across cell membranes. So the second law of thermodynamics says that the entropy, or the randomness of our universe, is always increasing, which means that particles or molecules move in a random fashion. So diffusion is defined as the movement of a molecule from high concentration to low concentration. So the molecule here moves across the membrane from high concentration to low concentration until it reaches what's called equilibrium, where it doesn't mean that there's no more movement. It just means that for every molecule that moves to this direction, we have a molecule that moves in this direction, the opposite direction, keeping it in equal balance. So diffusion is here on the left corner. Molecules start out in high concentration and they diffuse outward from high to low concentration. A good example of this is if you use like a body spray or a perfume or a cologne, like take Axe body spray and you spray it. If you spray it in one location of a classroom, eventually everyone in the class, if you spray enough of it, will begin to smell it despite where they're located in the class, because we know the molecules spread out and you detect those molecules at different distances. Now, the difference between diffusion and osmosis is that osmosis refers specifically to the movement of water across a membrane from an area of high concentration to low concentration. Diffusion means any molecule moving other than water. So if we're talking about a cell, we can't have an impenetrable boundary that doesn't let anything in or out, because all cells need nutrients and they produce waste products. So we need to get nutrients like carbohydrates, sugars, proteins, amino acids, lipids, and salts, as well as oxygen and water into the cell. And we need to get waste products like ammonia, salt, carbon dioxide, water, and products that are made by the cell to actually leave the cell. So this is the importance of having a membrane that is semi-permeable, allowing certain things in and out. So how do molecules get across membranes? So if this is the inside of the cell and down here is the outside, some of the molecules can pass directly through the membrane and some need to be transported. Which ones cannot get through? Well, polar molecules like water. Water can't just pass between the phospholipid bilayer. It has to go through special proteins called aquaporins that allow water to enter and exit the cell via osmosis. Ions cannot get directly through, so salt and ammonia and so forth have to be transported, and large molecules have to undergo a special process called endo and exocytosis. So in looking at this, we have transport proteins that allow specific molecules to pass through them to get into or out of the cell. And that's called facilitated diffusion. So when you use a protein to pass a molecule, it's going to be facilitated by that protein. Therefore, it's called facilitated diffusion. There's also active transport. Active transport means you have to actively put energy into the system. And in cells, energy is in the form of ATP. So that's like cash money for cells. And so you can also transport against a concentration gradient from low concentration to high concentration using a molecule of ATP as your energy source to power that like a pump. In looking at this, ATP is going to be required to move these molecules. So we have passive transport through, simple diffusion and facilitated. These do not require energy. And we have active transport, which requires energy. But what about substances? So this is just more explanation of that. What about substances that are too big to fit through proteins? So some things are just too large, and we have to go through a separate process called endocytosis and exocytosis. Endocytosis refers to substances moving into a cell, and there's two types, phagocytosis and pinocytosis. Exocytosis refers to substances exiting the cell. And the process, if you're looking at phagocytosis, you're talking about solid matter that gets enveloped by the plasma membrane and brought into the cell in what's called a little vesicle. You can also have pinocytosis, which means liquid. So like Pinot Noir, Pinot Grigio, those are types of wine. So those you can help use that term to remember. Pinot means liquid. So liquid can also be surrounded by the plasma membrane and brought into the cell. We can also have cell receptor mediated, which means that you have binding proteins that say, okay, this is a specific targeted molecule we're going to bring in. Endocytosis is enveloping in a membrane, meaning the plasma membrane gets shorter. The particles get packaged inside a vesicle, and that vesicle enters the cell. Water, however, has a special circumstance because it moves through aquaporins in a process called osmosis. We have three main settings for water. Hypertonic, which means not very much water, but lots of dissolved solutes. Hypotonic, which means lots of water. And isotonic, which means equal amounts of solute and water on both sides of a membrane. And we can talk more about that, but an example, if isotonic is like contact solution, you don't want to just put pure water in your eye. It'll be painful. You actually have some dissolved salts and substances in there. So saline solution, which is used in hospitals for IVs, it's also used for contact solution, has an isotonic or an equal concentration of solutes and water that the body has. So when you're talking about the direction of movement, in a hypotonic solution, hypotonic means there's lots of water in the surrounding environment, more water in the surrounding environment than inside the cell. Water will move into the cell. In the opposite case, a hypertonic environment, the solution around the cell has less water, so water exits the cell, moving from high concentration to low concentration via what's called osmosis, and it shrivels the cells up. So this is what causes plants to wilt, and this is what a plant should look like down here, and an animal cell should look like with a balance of water. I'm going to run out of time in this presentation. I know I've been talking pretty fast, so let's do this. There's a few slides left, and I'd like you to email me or use my contact form, and we can talk about the remaining slides for this to make sure that you understand the key concepts.